Okay, everybody, welcome to yet another episode of our Western Biology Evolution Seminar Series, or WBESS for short. This is, in fact, our third episode in season 2. And I must say, I am very excited with this project, with the way things are going, and I've been learning a lot about evolution. So I do hope you have all been watching this series along with me. Today, I have the pleasure to have Sarah Santos with me. Sarah is a PhD student here at Western. She's also from Brazil, just like myself, and she has been studying different mammal groups during her graduate trajectory. And yeah, I will let her introduce herself and take it away. So Sarah, I'll be in the background, and I'll be back with you when it's time for Q&A, okay? Take it away, Sarah.
Okay, thank you for the introduction. So yes, I've been working on hybridization using whole genome data, and I did that in my master's and my PhD. So I'm a PhD student in the Coltman lab, the David Coltman lab. And today I'll just talk about hybridization and how we can detect hybridization with genetic data. But before we go into those details, we need to focus a little bit on what is a species tree and how species evolved over time and diverged and their relationship to each other. So a species tree basically will represent the true or the original speciation that happened between the group. And we can use different types of data to understand their evolution. So for example, we can use morphological data, we can use biochemical data, and we can use molecular data, which is basically the genetic or the whole genome, the complete DNA of those species. What we have seen for different groups, specifically for mammals, is that depending on the data that we are using, we can observe different signals. So for example, we will observe the species tree, but we can also observe other types of relationships. So instead of, for example, having C and D being closely related, we would have C and B or D and B species. And so when we look into the complete DNA, so the whole genome, what we see first is if we analyze the complete version, and typically the majority of the genome will represent the species tree, but more specifically, we have to look or analyze smaller pieces. And typically when we analyze smaller pieces, we have the species tree there in multiple places. So the majority of the genome will represent that topology or that tree. But we can also observe other relationships, as I mentioned before. And typically we call those alternative topologies or alternative trees. And they basically would just represent a different evolutionary relationship. And that can be caused by different evolutionary forces or different evolutionary mechanisms, what we call then genealogical discordance. And genealogical discordance has so many different causes. One of them can be introgression. And introgression happens when hybridization happens. And here I just have an example of 3 species of birds in South America. And let's say we had species A being a dark pelage or dark plumage bird. And then we have species B being white and species C just having a different tail, but also being white. And then they have diverged, they are their own separate species, but then for some reason, some geographic distribution, some type of barrier that doesn't exist anymore. Those 2 species, so A and B, have what we call secondary contact, which basically is they hybridize. And when they hybridize, they can generate hybrids. And in this case here, this example, we would say that now the hybrids have a gray feathers. And then for introgression to happen, so introgression is when different pieces of genetic data get introduced or reintroduced to a genome. For that to happen, then we need the hybrid, so these new specimens, to hybridize with the original species. So let's say a hybrid would hybridize with species B and then introduce new genetic information into their genomes. And how we see that is sometimes, of course, we'll have an adaptation that happens. And so in this case, let's say that the hybrids hybridize with species B and introduce this new feather to their head, so new color. So now they have gray heads. And that somehow contributes to their adaptation to the environment. But of course, not always. We're not always able to see that in phenotypically. We don't have that characteristic. But sometimes it will be something internal, like something related to digestion or something behavior. But in this case here, we just have the different color. And how that translates into the genome is that we'll have pieces in species B that they're also present in species A. Because of that hybridization process and because of how integration happens. So we now have some type of genetic information in species B that is also contained in species A. So that's how integration happens, and that's what I study. We have cases of recent hybridization that has happened between different species. I'll just give you an example before I talk about some of the results that I have. But recently, a hybrid between polar bears and brown bears or grizzly bears have been found. More specifically, I think there are around 8 individuals, and they're all coming from like the same more or less region, and they're called growlers or pizzlies, depending on who was the male and who was the female. So that's something that we see nowadays, and something that we typically before in time, we thought that hybridization between mammals was very rare, but now we see that it's not that rare, and that is also evidenced in the genetic data. And so when we look more specifically at the genetic data, we try to analyze that genetic data. We see that there are a lot of hybridization signals between different bear species. So for example, I mentioned polar and brown bears, but we also have between American bears, Asiatic bear, sun, and sloth bears. And that signal is from past hybridization events, not necessarily just recent. Because of course, some of them don't have overlapping distributions nowadays. They had maybe back in the day or thousands of years ago. But that signal is still reflected in their genetic composition. And it's just another example, because I mentioned that we can have morphological data, we can have biochemical, and we can have molecular data. But also when we're looking at the nuclear data, or the mitochondrial data, we can have different relationships being observed on those as well. And so here's just another example where we have brown bears and polar bears. The majority of their genomes, they're separate. They are their distinct species. But then if we look at the mitochondrial DNA, we have the polar bears clustered within the ABC Islands, which are in Alaska. And so that reflects on the fact that they hybridized in the past. And we can still observe that in their mitochondrial DNA. But also, when we are talking about hybridization, so I mentioned recent, something that that happened because of climate change, and then their distributions starts to overlap again. But we can see some of the ancient past thousands of years that are also reflected in the DNA data. And we can also try to fill in the gaps when we include ancient DNA. So if we include fossil data, and of course, if we are able to, because fossils can be very rare, can be very degraded, so it's sometimes hard to get DNA from that. But if we are able to, we can kind of fill in the gaps that we wouldn't be able to with only the contemporary DNA. So here's just another example that cave bears contributed around 0.9 to 2.4% of the genome of grizzly bears. And so just another example of how hybridization has shaped the DNA of different species, specifically here of bears. But in my presentation, I'll focus more on the big cats, so the Pantera, which are 5 species, and the wild sheep, the North American wild sheep. And to analyze hybridization patterns, one way we can do that is by having the complete genome, and then we can have what we call a reference genome. A reference genome can be, for example, a domestic sheep, some very well studied group, and we can get our data for different individuals and map that to the reference genome. And so that we have every individual having the same coordinates. So that's what I'm showing here. They would just all have been aligned to the reference, and then we can analyze and we can partition the data and use different methods to understand their evolution. So one of them is we can use a PCA just to look at genetic similarities. We can also look at phylogenetic trees, as I mentioned before, and try to distinguish between a species tree, an alternative tree. We can use more specific methods to estimate intergression or hybridization. And so just to exemplify here what we get from those types of analyses with the PCA, we can try to delimit the species and their clusters. So like the colors here would represent different species potentially. And then we can also try to detect potential hybrids between these different species, which would be, for example, here, this red dot between the yellow and the red populations. So that's one way of visualizing the data. This is just an initial screening way of observing, of detecting different species and different hybrids or potential hybrids. Then we can also use phylogenetics or phylogenomics when we're using whole genomes. And basically, we'll just either look at genes specifically, or we'll look at the genome partitioned into smaller pieces. Those pieces can be of different sizes. They can be bigger or smaller, and I'll talk about that later on. But then basically, we're just estimating the evolutionary relationships and trying to distinguish between a species tree and an alternative tree. So the original speciation and genealogical discordance. So just to recap that, intergression is just one of the genealogical discordance patterns that we can observe in the genome. And then we can use more specific methods that estimate intergression or hybridization. One of them is d-statistics. And d-statistics is basically trying to find patterns of ABA-BABA. And here we have A as in representing ancestral. So it's the ancestral state. So for each one of the windows that we analyze in the genome, we have the sites, the nucleotide sites. And for each one of those, it will compare for all the 4 species that you're analyzing. It will try to come up with either A or B. So A for ancestral and B for derived or for intergression. And then it will try to look at each one of those sites for all the species and come up with that pattern and use statistics to determine whether you have, for example, here we have ABA. So B and B here would place species 2 and 3 as sisters. And then BABA, where we have 1 and 3 as sisters. So it's just different from the species tree. We need to know the species tree for this specific program, but it's different from the species tree because it represents the intergression signals. And it does this with the ancestral state. So I mentioned 4 taxa or 4 species here, because the 4th species will be the outgroup. So just basically something that is not related to that intergression event. And that's trying to establish what is exactly the intergression. And it only happens between species 1 and 3 or 2 and 3. And then it will do this pairwise intergression estimation. And then we can also use more specific models of evolution. And basically here we'll try to have our species tree, of course. And so here the thicker lines or the solid lines are the speciation history. And then we can look for specific hybridization patterns. And what admixture graph does is you will come up with this model. You have your species tree and you have the hybridization patterns that you want to analyze. And then it will take into consideration the data you provide, so the genomic data that you provide and each one of the sites. And it will come up with numbers that will represent patterns of hybridization. So in this case here, we would say that the Yukon, or the Yukon sample contributed 70% of the Wyoming genome, and then Bighorn would have contributed 30% of that based on the model and the data that was provided. And so with that, I'll now talk about some of the findings that we had with big cats, more specifically. And big cats, I studied them during my master's in Brazil with my supervisor was Eduardo Eiserick. And these were all the people that were my co-authors or that contributed with samples, with analyses for this project. And here we estimated intergression among jaguars, lions, and leopards specifically. And we have 5 Panthera have been extensively studied in the past. And here is the species tree, or what we thought was the species tree. But even before this tree here, there were a lot of studies with morphological data, biochemical data, molecular data that would have this different trees representing the true speciation or the original speciation. And here in our paper of 2017, we observed that lions and leopards would be sister clades, and then jaguar would be outside of that pair. So that would be their true speciation. And then we would have snow leopards and tigers as sisters, and then domestic cats as the outgroup. And they would have evolved around 4 million years ago, and we would just observe signals of hybridization between different combinations of these species here. But this would be the species tree. But when we analyze the whole genome, and again, partitioning it into smaller pieces, we often observe that what we call the species tree were placed in higher recombination rates, whereas the non-species tree topologies were placed in lower recombination rates. But then because of how the majority of the genome represents lions and leopards as sister clades, we thought that was a species tree. And then we analyzed patterns of intergression between different combinations, and we observed, for example, signals of hybridization between jaguars and lions, and between lions and snow leopards and tigers. And so just different combinations of integration signals. And in another study, a 2019 study, they observed again that lower recombination rates did not retain the majority, or the tree that represented the majority of the genome. So even though lions and leopards are sister clades, they're not placed in lower recombination rates. And that's very important, because when we have high recombination rates, it's easier to retain integration, because let's say we have a big piece of integration of DNA that was integressed. It's easier to be placed in a part of the genome that can break it up and introduce that information than when we have a low recombination region where all of that would have to be placed there instead of just breaking it up into smaller pieces. And so because of that, my project came into perspective, because we wanted to analyze the phylogenomics using different window sizes, so different fragment sizes. We also wanted to use other types of methods, such as the statistics, and we also generated models of evolution to try to visualize what was the species tree and what we could tell from the genomes using multiple individuals per species. And so the first thing I did was to generate different window sizes. So we had from 50,000 base pairs to 5 million base pairs in the genome. So just different fragment sizes throughout the genome for different chromosomes. And we had our main topologies, which were either the topology 1, which is the majority of the genome, placing lions and leopards as sister clades. Topology 2 as jaguars and lions now being sister clades, and topology 3 with jaguars and leopards. So just the main topologies there. And as I mentioned, we had a recombination rate throughout the genomes for the different chromosomes. And the most common topology was topology 1. So lions and leopards are abundant. The relationship is abundant in the genome. So that's the yellow color here. And for the different window sizes, that's the signal that we obtain. And then the second most common is the topology that places jaguars and lions instead. And then topology 3 is the least common of all three. So as I mentioned, for the different sizes. And if we look specifically for that signal in the X chromosome has what we call a code spot in the genome, a code hotspot, sorry. So that's this region here, which is composed of very low recombination rates. We see that topology 2, so jaguars and lions, are placed there. We also have some of topology 3, so jaguars and leopards. But topology 1, which is the majority of the genome, are placed in higher recombination rates, independent of the chromosome. But here is a very specific example of that. And so they're just placed in these higher recombination rates compared to 2 and 3, which are in lower recombination regions. So just really confirming what we had previously established, but using different window sizes across the genome. Then we also estimated the statistics, so the Abba-Baba patterns. And now we try to establish that the species tree is not lions and leopards. So we ruled that out, and we generated these statistics using the 2 new possible species tree. So now we had either jaguars and lions as sister clades, or jaguars and leopards. And the numbers here are just different combinations of individuals that I had. And then the arrows represent introgression signals, so bidirectional introgression signals. We just don't know the direction of the donor to the receiver, but we know that the signal is present between these species. And then we had a great number. So this number here represents the windows, so the windows throughout the genome. And we had 8,500 windows or fragments that represented a relationship, an introgression relationship between lions and leopards. So that's very striking, and it's not really something that we expect. As I mentioned before, the species tree is typically, what we expect is that the species tree will be the majority of the genome, but here we have the opposite. We have that the majority of the genome represents hybridization. So that's very striking and very different. We also ran some models of evolution, and those models also ruled out lions and leopards as sister clades. The only thing here is that we cannot distinguish between these 2 trees. We cannot identify which one of them are the true speciation history, but we know for a fact that the topology that we thought was the species tree, so lions and leopards, are not the true speciation history. And so just to sum up what those results represent here is the possible story here is that the ancestors of lions, leopards, and jaguars originated in Asia, and then they dispersed to Europe, to Africa, to the Americas. And of course, we have some records of fossil records of lions in the Americas. We have jaguars, or living jaguars in the Americas. And then we also have lions and leopards in Africa and Asia. And so it's very likely that after they speciated, after we had the original speciation happen, lions and leopards just had this overlapping geographical overlap that allowed them to keep on hybridizing throughout their evolution, and that's why we see the genome being overwhelmed by introgression. And so that's very different from what I'm going to talk about when I mention wild sheep, because wild sheep we have a more simple case where the majority of the genome really just represents the species tree. So with wild sheep here, that's my project for my PhD, where I have David Coltman as my supervisor. And in this project, we have both contemporary and fossil record DNA. So the first part of this presentation, and I have summarized some of the results that I found. I'll just mention the living or the contemporary sheep. Basically, we have thinhorn and bighorn sheep as the North American species. Thinhorn has also 2 subspecies, Doll and Stone. As you can see here, Doll, it has this white coat color, and then Stone is darker, which is more similar to bighorn sheep. But they're different species, so Stone and bighorn are different species, but they look more similar than Stone and Doll. But as the name here shows, thinhorn sheep have this like thinner horn compared to the bighorn. So there are different species there. They also do not have current overlapping distribution. So Dall is more to the north in Alaska, Northwest Territories, and the Yukon. Then stone sheep is in British Columbia and overlaps with Dall along the Yukon border. And then bighorn sheep is across western US and some parts of the Rocky Mountains in Alberta and BC, but again, not overlapping with stone currently. And in the past, lots of studies have proposed that stone and bighorn sheep have hybridized in the past. That was shown with mitochondrial DNA data. And what happened here, or the theory is that in our galley, so our galley is another wild sheep, but in Asia, that is placed in Asia, snow sheep as well. And snow sheep, thin-horn sheep, and bighorn all form the Paxidiform sheep. So they're all very closely related. So outside of here, of this Dall, stone, and bighorn, we would have snow sheep. And so basically the theory is that an ancestor, either snow-like or galley-like, entered the Americas here and then diverged into the thin-horn clade and then the bighorn. And within the thin-horn, we would have Dall, a Dall-like ancestor that was either potentially caused by a mutation followed by natural selection, because it was probably an adaptation to the environment. And then we could either have a Dall-like ancestor that became a stone after hybridizing with bighorn sheep, or we can have the ancestor of Dall, so the thin-horn here, that would hybridize with bighorn. So in either case, hybridization would have maintained this darker coat color, or reintroduced the darker coat color into the stone lineage. And when we analyze the mitochondrial DNA, as I mentioned, that signal is present. So what we see is that bighorn, we have the distinct clade. So we have Dall here, and then we have bighorn here, but then instead of stone being here, we have some individuals being placed with Dall and some individuals being placed with bighorn sheep. So the signal of hybridization is present both in smaller pieces, so the control region of the mitochondrial DNA, or the complete mitogenome. So we had an undergraduate student work on that, and she observed that the mitochondrial DNA, the complete mitochondrial DNA, has that hybridization signal. And so with my project, then we wanted to look at the complete DNA, so the whole genome, and analyze different patterns of integration throughout the genome. We published this in molecular ecology, and we used multiple individuals, multiple genomes. Again, we use a reference genome, so we use the domestic sheep, and then we map those to the domestic sheep, and we analyze different patterns. So we generated phylogenomic trees, and we generated these statistics, so the ABABABA patterns again. And in this case here, we wanted to look for the species tree. So of course, thin-horn sheep, Dall and stone should cluster together in the majority of the genome because they are the same species. They are just subspecies of thin-horn sheep. And then we would have bighorn outside of that as a sister clade. And then we use the domestic sheep as the outgroup. But we also wanted to look for alternative topology. So we wanted to look at the relationship between bighorn and stone and bighorn and Dall. So alternative topology 1 and 2 compared to the species tree. And so we generated different data sets depending on the combinations of individuals. We generated different sizes of genomic fragments and different step sizes as well. Because as I mentioned before with the big cats, we have recombination rates that can generate different signals in the genomes if they are higher or lower recombination rates. We didn't have that information for the wild sheep. And so we use spaces between the windows to account for that. So instead of having the genome and then just partitioning, we had spaces between those partitions. And so here what we observed is that the majority of the genome shows the species tree. So for example, we have doll and stone as sisters, and then bighorn outside of that. We also included snow sheep, but snow sheep was another outgroup. So it was just to really dissect the relationships between thin horn and bighorn sheep. And so more than 50% of the genome represents the species tree, followed by stone and bighorn sheep. So we have them as sister clades, around 30% of the genome. And then around 19% of the genome, between 16 and 19, we had doll and bighorn sheep. But these phylogenomic signals here, the program doesn't really account whether it's hybridization or other types of mechanisms. And so we use more specific methods to estimate hybridization. And so one of them was a model of evolution called PhyloNet, which is based on those trees that we generated before. And here what we observed is that stone sheep would have 71% coming from doll sheep and 29% coming from bighorn sheep. So it had that hybridization signal with bighorn sheep, and this was the best fitting model that we observed. And this number is important, and we'll keep that in mind for the next part of my talk with the fossils. But we also estimated these statistics. And here again, the bidirectional signal between species, we observe far more number of windows representing intergression between stone and bighorn than doll and bighorn sheep. And so just to summarize this part, we use different data sets, different combinations of analyses of windows or fragment sizes. And we have a robust signal that represents, the species tree. So we have doll and stone as sister clades, and then bighorn outside of that clade. And then we also have a relationship between stone and bighorn sheep that either maintained or developed the darker coat color in those fragments that had intergression between stone and bighorn. Oftentimes, coat color genes were present, and so it's very likely that intergression contributed somehow to that coat color, the darker coat color in stone. And then we also had a less frequent relationship between doll and bighorn sheep, or less frequent compared to stone and bighorn. And one possibility for that is that if we consider that stone is right in the middle of doll and bighorn sheep, it could have hybridized with bighorn sheep, or the ancestors could have hybridized, and then the ancestor of stone would have hybridized with doll, or even currently hybridizing and passing along that genetic information. That has been proposed for bears. So grizzly bears have a wide distribution, and what has been proposed is that grizzly bears would have hybridized with polar bears and then hybridized with American black bears, passing the information from polars to American black bears. So that could have been the case here with doll sheep. But we can also think that maybe we're just missing a lineage. We're missing a fossil record here. And that's another part of my project where we used ancient DNA to analyze patterns of hybridization. And here I just wanted to exemplify a little bit of how that works. So we have either a teeth or tooth sample. We can have different types of samples, but in our case, we had tooth or teeth samples. But here we could have a skull. We could have a petrosal, which is a bone in the ear that has very preserved DNA. And we need a very specialized lab to extract those DNA. So it's very, there are a lot of protocols that need to be followed. There's a lot of worry about contamination. So just to exemplify how that looks. And here is the natural trap cave in Wyoming, where one of my samples came from. This natural trap cave is known to have a rich amount of fossil records. And so just to exemplify where the fossil record comes from. And more specifically, we had 4 main samples in this project here. 3 of them are from the Yukon. So the skulls are from the Yukon. And then we have 1 from Wyoming, so the natural trap cave. And Here again, we had a contemporary sample, so doll here in Alaska, Northwest Territories, and then we have stone sheep in British Columbia, and we have bighorn sheep in Alberta, Canada, and then also Montana in the US. And our fossil samples were from the Yukon, 3 of them, and 1 from Wyoming. And again, we used a reference genome, so we used a domestic sheep, and we mapped all of the living and the extinct specimens to that domestic sheep, and we used their complete genomes to estimate genetic similarities with BCA. We also estimated models of evolution, different models of evolution, and we got the best fitting model. And we estimated different combinations of individuals with these statistics to detect patterns of integration. For the PCA, we did different types of filters because it's very important to filter the ancient DNA to make sure that we don't have contamination, that we don't have errors in the sequences and those types of problems. And so we had different filters. We had what we call a lenient filter, which is a single read approach. And basically what that does is for each site in the genome, it will randomly select one base and one of the bases, the nucleotide bases, and then we'll generate a PCA based on that. And that was just to keep a little bit more of genetic information and see what comes out of that. And then we also have had a stringent filter or filters that that included a bunch of steps that would allow us to really have a true signal for the ancient DNA. And I have the 2 of those plots here. And then the rest of the analyses we use in stringent filters. And so with the contemporary species, we have stone up there and then doll. We also had bighorn down there. So again, the stone and doll are their own species, so thin-horn sheep. And then we have bighorn as a separate species. We had snow sheep very distant. So the Asian Paxiform is distant from thin-horn and bighorn sheep. And then we looked specifically for the fossil record. And the Yukon samples, they cluster with stone and doll, which is expected given their distribution. But the Wyoming sample, which was a bit striking, was placed between bighorn and thin-horn sheep, which means that it is a potential hybrid lineage. When we look for, when we use stringent filters, again, we observe thin-horn sheep separate from bighorn and separate from snow sheep. But then we also see that the fossil records are closely related here. So the Wyoming is closer to the Yukon samples and closer to thin-horn sheep, which is a bit different than we were expecting since before we had that hybrid lineage. But these, for the PCA, we filtered a little bit, but we filtered it even further for the other analyses. And then when we try to generate models of evolution and try to generate patterns of these statistics or ABABABA, we saw some very interesting results here. So I'll just walk you through the model of evolution together with these statistics, because it's a bit easier to look at phylogenetic trees than it is to look at networks. So we had snow sheep as the outgroup, so the least related to all the other species. And we had a branch that diverged and generated different lineages. And here, what we have next is one that generated the living species and some of the fossils, being doll, stone, and bighorn, and another one that would represent a ghost lineage or ghost lineages. What that means is we have a missing piece in this puzzle. Basically, potentially we have a missing fossil record that we did not sequence. And then we had another divergence, so they diverged again, and that would generate the thin-horn sheep and the Yukon. So the Yukon would be outside. It would be less related to the contemporary doll and stone. And then we would have another branch that would lead to Bighorn and Wyoming sample, which makes sense given their distribution, given the distribution of contemporary Bighorn, that Wyoming would be a sister clade. But then we have some signals of hybridization. And like before, and I mentioned this number before with Pilonet, we had that 74% of the stone genome would come from the Thinhorn clade, and then 26% comes from the hybridization event with the Bighorn clade, or with the ancestor of Bighorn sheep, which is a good thing because it confirms previous results. And then we also had something different here, where we have the Wyoming sample having 35% coming from the Bighorn lineage, but also 65% coming from this missing fossil. The Yukon sample also had 37% coming from the ghost lineage and 63% coming from the Thinhorn clade. So that's also reflected here in the D statistics. If we look for, if we look for stone and bighorn sheep here, the 26% makes sense with the bidirectional relationship here. And then if we look at stone and Wyoming, or Yukon and Wyoming, the missing piece, the missing fossil, could be represented there, because this signal here was very weak. And so it really means that these statistics just can't account for missing fossil, missing lineage. And so it's very interesting to see that the model actually is able to detect such patterns. And then we also see 6% coming from the ghost lineage into the Dahl clade. And of course, Dahl is a true representative of Thinhorn sheep. So 94% of its genome is a Thinhorn, but then it has this 6% coming from the ghost lineage. And when we look at the D statistics, we observed that Dahl and Yukon, or Stone and Yukon, had hybridization signals. So again, that missing piece could explain some of those signals in the D statistics, since they can't estimate those results. But here in this study here, we sequenced for the first time the Pleistocene wild sheep, and we observed different patterns of hybridization. So we observed stone and bighorn again. Their pattern is very clear, but we also observed a potential hybrid lineage with the Wyoming sample. But of course, this sample here was had less DNA, was more degraded, and so of course some of that could be contamination, or some of that could be some type of missing information that we don't have. But the ghost lineage is still present and it's still possible. We tried that model, we tried running models without the Wyoming sample, and the ghost lineage or lineages are still present. And what we expect is that the missing piece is older than all of these samples, and would explain, or better explain, the hybridization that happened between these species. And then the Yukon fossils are true representatives of Thinhorn sheep. So with that, we can conclude that different patterns of hybridization can lead to different can be represented in different forms in the genome. So we can have either the majority of the genome being overwhelmed by hybridization. We can have more specific hybridization patterns, such as stone and bighorn sheep, and we can have something ancient that is a fossil that will either may lead or help us better understand hybridization patterns, but it can also tell us if something is missing, and that we can better understand their evolution. And with that, thank you for your attention today, and I hope you enjoyed the talk.
Thank you. Thank you, Sarah. I'm pretty sure everyone's going to enjoy your talk. This was very insightful. Thank you. So I do have a few questions, and I think my questions, we can group them in two categories, right? So the first type of question that I have are more technical, I would say, more detail-oriented, and the second type of question is more conceptual. And both of those categories come from a place of curiosity, really, because although, you know, I was able to follow your work and talk along, and I did understand most of it, it's not really my field, so I do have a few basic questions, let's put this way. So in terms of the technical questions, there was a moment in which I think you mentioned that you did not have the recombination data for the sheep genomes in comparison to the big cat's genomes. Is that right?
Yeah.
So uh, so what would be this recombination data be like? And we don't have it because the sheep genomes aren't, you know, assembled at the chromosome level, or what is it like? Just out of curiosity.
Yeah. So that's a good question. So for the big cats, the domestic the domestic cat has been extensively studied, and so they generated the recombination rates, and they have been updating that for the genome assembly. So that's how we have that information. But for the wild sheep, there is one for one specific assembly. But for the newer assembly, which is the one that I used, they didn't have that information. So that's why I didn't use it. It's just that I wanted to use the newer version because it was better assembled. It had the chromosome level. But that's mainly why. It's just that they're just studying different aspects of the genomes of sheep versus big cats.
And again, just out of curiosity, so I remember seeing in your the first half of your talk when you showed us the genomes of the big cats, and there were, I don't remember the precise number of chromosomes, but there were different types of chromosomes. I think there was, let's say, chromosome A 1, 2, 3, B 1, 2, 3, something like this. And what does that nomenclature mean? Yeah, if you could explain to us on that.
It's just a different type of label. For example, like the humans, they just do 1, 2, 3, 4, and so on. For the big cats, they did A1, A2, and A3.
Oh,
Then
okay.
they kind of did like the alphabet.
Oh, okay.
Yeah, it's just it's just
Okay.
a different type of label. Yeah.
Interesting. Um, Now, speaking of the ancient DNA, as you showed us, and you mentioned very well that, you know, we should be careful with those analyses. We have different, you know, filtering stages. And then when we're talking about whole genome analysis of ancient DNA, I think what we mean with whole genome analysis with that material is that we're analyzing the entire genome that we can have access to, right? But we are not able to assemble entire genomes as of yet from ancient DNA, right? Because the DNA itself is fragmented and mutated and things like that, right?
Yep. Yep.
Yeah,
Yeah.
just trying to hammer those concepts into the viewers. Now, moving to the more conceptual questions, I think the concept of, you know, hybridization, introgression, I find them fascinating because I think they challenge our you know, limited view of how species evolve and how species interact with one another, right? One thing that I'm always curious, and I would like to hear from you, how population genetics data can either inform introgression studies or can be gathered from introgression studies. Because, for instance, we're thinking about, you know, the big cats, wild sheep, the bears you've talked about. Sure, we can find those signals of hybridization depending on where we look at the genome. But I'm thinking about the actual hybridization event, right? So because hybridization, especially in those, you know, big mammal species, those species, I might be wrong, you correct me if I'm wrong, but, you know, those species are often, you know, they have like wide ranges, they might be solitary, they don't have that many encounters, right? It's not like birds like flock together, go to their breeding grounds. So what I'm going to is that how frequent a hybridization must be for us to detect its signals, right? Because if you have like one bear, one polar bear breeding eventually with a grizzly bear, right? Only once, that signal will be lost, right? So I'm just trying to connect, you know, population level information, right? Population dynamics with the actual genomic signatures. Do you see what I'm saying?
Yeah, I see. That's actually something that happens or happened with the polar and grizzly bears, because we have both captive and wild hybrids, right? But even though we have hybrids in the wild, they're not. It's not like they're hybridizing all the time, and a lot of hybrids are present. There's a recent study where they found 8 individuals, if I'm not mistaken,
I see.
and it's just those 8 individuals. They don't have more individuals that are hybrids. And so I think there needs to be more overlap, and there needs to be more chances to, and it also means how diverse they are from each other, how likely it is that they will seek one another, because of course there will be behavioral aspects, there will be reproductive or sexual selection aspects that would maybe not allow them to interact as much compared to before. I think with the past hybridization, it probably likely happened while they were diverging, and so they didn't have a lot of those barriers, even geographically or just behavior or other characteristics that would allow them to reproduce or seek one another, if that makes sense. Um,
And I'm thinking, I agree with you, what you said makes sense, but I'm thinking as well, as these, hybridization events happen, they might be followed by founder events as well, right? Because I'm just thinking of Big Ship, for instance, right? Which, to my understanding, it is relatively, I mean, they herd, right? They have herds, but they're not that, they don't have big herds, right? They're more solitary, aren't they? Especially the males, the sheep, or not?
The sheep, it depends a little, yeah. I've seen some, yeah, they're not like super big for
Or they
groups,
have
no.
herons there. I'm totally ignorant on those guys. But I'm just thinking, perhaps like a hybridization or a few hybridization events have happened, and then you have, um, you know, either by chance, just those that carry the hybridization signals survive and leave offspring, or you have, which inevitably is a bottleneck, right? Effect. There's a bottleneck effect, or a founder event in which a subpopulation goes to another location. Yeah, again, I'm just in my mind, I'm just trying to, come up with reasonable stories. But again, um, I think the beauty of evolution is that evolution is a historical discipline by its own nature. So, as much as we can try to dissect the signals of what has happened, we can certainly, I guess, I feel we can get closer to what has happened, but we will never be really certain as to what has happened, right? So many things
Yeah.
must have happened. But I think that's the beauty of evolutionary biology. We often need to be content with uncertainty, right? Uh,
Yeah, yeah, I agree. And I agree that genetic drift will for sure maybe change a little bit of that genetic composition, even if hybridization happens. I think there needs to be more hybridization for the signal really to be maintained, and then we would be able to see that. And I think what we study like from the past might help us with like with climate change and everything changing, and just like maybe be able to better understand and even better mediate like future hybridization patterns, but I think we're still not there yet,
mhm.
maybe. But we'll probably get there eventually with climate change.
Oh, absolutely. And also, I mean, again, life history traits, they really dictate how prone a particular species or particular groups of species are to hybridization, right? Uh, let's talk about plants, for instance. It's hybridization all the way. So I think life history traits really dictate the probability of finding those signals in the genome, how profound and widespread they are, and things like that. And all this time I've been thinking about microbes, for instance, right? And if you think about prokaryotes, for instance, we don't even talk about the true species tree anymore, right? We talk about networks. Some people say, oh, we don't have a tree of life, we have a bush of life for prokaryotes. So yeah, I think these things, these ideas and concepts are important for us to think about, to enrich our understanding of evolution as a whole. So yeah,
Yeah.
thank you very much, Sarah. This was great. I'm pretty sure, again, folks at home will enjoy your talk. And I'm hoping that, when they watch your talk and if they get interested in your research, they will reach out to you or to your supervisor, David Coltman, and conversations can get started from there. Okay.
Oh, yeah, for sure. Thank you again for the invitation.
Thank you. So I will now stop the recording just a second.